We're just a little over 4 o'clock, so I think it's good for us to get started. Let me introduce Lyle Muller. Lyle was actually a Brown undergrad. He received his degree from neuroscience with honors. He then went on to Paris, best city in the world, to get a master's degree from École Normale Supérieure. In computational neuroscience. He did his PhD advised by Alain Destex at Pierre Marie Curie. And then he went on to work at the Salk Institute with Terry Sejnowski for 5 years as a postdoc. And he has been an assistant professor in the Department of Mathematics at Western University in Canada since 2019. Today, I'll talk about traveling waves in Cortex, spatiotemporal dynamic shape, perceptual and cognitive processes. Please join me in welcoming Lyle.
Thank you so much. First of all, I wanted to say thanks for the opportunity to visit Brown and to meet everyone here at the Carney Institute. And as introduced, I'm Lyle Muller. I'm in the Department of Mathematics at Western, where I moved after postdoctoral research with Terry Sejnowski at the Salk Institute. So just to get started, my lab brings together neural data, network models, and theory, where the idea is that we always keep close to experimental data, starting by developing new computational methods to analyze cortical dynamics. We then connect this analysis to computational models that allow us to infer more and capture the network level mechanisms behind what's observed. And we finally allow the model to help us suggest new analyses and further experiments. And now the general idea we have for the neuroscience we're doing is how do the single trial and moment by moment dynamics of cortical circuits affect sensory processing and behavior? And what we've increasingly found by considering these single trial dynamics over the past few years, and what I'd like for you to take away from this talk today, is the importance of considering cortical dynamics in both space and time. So I can explain this point a little more by providing an analogy. So we often observe neural dynamics at a single point, as in this zoomed in movie of a flock of birds, where we appreciate that the local dynamics have high disorder. But when we zoom out to a much larger scale, here now seeing the whole flock, we see that sophisticated order is present. In what I'll show today, the individuals here will represent spiking activity at a microscopic scale, and the waves sweeping through the population will represent sparse waves of neural activity propagating over the entire brain region. So as a motivating example here, in neuroscience, we're generally interested in the flow of information across brain areas, as for example, evoked by a small visual stimulus. Stimulation with a small point of light creates activity in primary visual cortex, or V1. Higher visual areas are then activated in rapid succession, fed by this activity in V1. But importantly, 80% of the connectivity in cortex occurs on the scale of 2 millimeters within an individual cortical region. It's just now become possible to image this scale in individual cortical regions with high spatial and temporal resolution. And we're interested in studying the dynamics there. So now if we focus on multi-unit neural activity in a local patch of visual cortex, as a small visual stimulus is repeatedly presented to the animal, we see that just after stimulus onset, or t = 0 here, the evoked spike train is highly variable from trial to trial. And that's even during the firing response in the first 200 milliseconds after stimulus onset. So on some trials, you get a lot of spikes, and on other trials, you get almost none. Now, if we take this small patch of visual cortex and look at its local field potential, or LFP, which is thought to represent the summed synaptic currents in its neighborhood, and can provide us a first measure of local excitability, in the average over many, many trials, we can see a clear negative deflection aligned to the multi-unit spiking response. And this stands clear of the so-called spontaneous or background activity before and after the response. So this is how we've traditionally studied stimulus-evoked responses in sensory cortex, but it can be somewhat misleading because if we look at the LFP in each trial, we now get a very, very different picture, where the spontaneous activity is now the same size as the stimulus evoked response. Today, I'm going to show that these spontaneous and stimulus evoked fluctuations of neural activity are not exactly random noise processes, but instead are organized into smooth neural traveling waves when you look carefully across multiple points in space. These waves travel across single cortical regions in awake animals and across the whole cortex during sleep. And because they can dynamically influence neural excitability, they may represent a previously unappreciated mode of neural computation. As a brief overview, this talk will be organized into two parts. In the first part, I'll show that spontaneous and stimulus-evoked traveling waves in visual cortex of awake monkey shape normal sensory processing. And in the second part, I'll show recent work from my lab asking what computations can be done with these traveling waves in visual cortex. And I'll say here that I think these are specific instances of this more general idea. That is, again, how do the single trial and moment-by-moment dynamics of cortical circuits affect sensory processing and computation? So first, we studied in detail the spatiotemporal dynamics in visual cortex during the awake state. And just to get started here, we can consider a small visual stimulus, which will evoke spikes in thalamocortical fibers leading to the input layer of V1, eventually driving responses in a restricted feed-forward driven zone in the superficial layers of cortex. Now we know that long-range recurrent horizontal fibers then carry spikes evoked in the feed-forward zone over a large extent, up to several millimeters of cortex in this response zone here. And now the question is, what's the basic spatiotemporal form of what's evoked in the response zone? Is this like a stationary bump of activation? Or is it something with more spatiotemporal structure, such as a radial wave traveling outward from the point of input? And I'll say here that while phenomena like this had been observed during anesthesia, people hadn't really studied the spatiotemporal structure of population responses at the single trial level and in the awake state, where activity is much more complex and responses are more difficult to analyze. But these distinctions are important because they lead to different types of coding in the visual cortex. For example, stationary bumps keep activity localized at a constant point in the retinotopic map. So here at each point in time, your point of maximum activation is at the same point in the retinotopic map. While traveling waves can dynamically influence neuronal excitability as they go across the retinotopic map. So here, the maximum point of activation spreads outward from the point of input. So to study spatiotemporal dynamics during visual responses, I began a collaboration with the lab of Frédéric Chavannes in Marseille to analyze optical voltage-sensitive dye recordings from the awake monkey. And now VSD is an imaging technique in which a fluorescent dye is used to stain the cortex, and it allows recording the local polarization state of small neuron groups with very high spatial resolution. So at left are the imaged cortical areas with primary visual cortex here. This is the 1 millimeter scale bar. And at right are the temporal dynamics of the population response to a small point of light presented for 50 milliseconds and then averaged over this V1 ROI. And we were interested in the dynamics across the cortex during this fast, transient onset response associated with this high contrast visual stimulus. And now when we look at the data and the way they're typically analyzed, after smoothing and averaging over tens or hundreds of trials, the stimulus responses we see look similar to a stationary bump. Where plotted in the movie I'll show is activity in primary visual cortex. So this crescent here will be represented as a crescent here in the movie. This is the 1 millimeter scale bar, and the VSD signal here is plotted in color with light colors indicating depolarization. Now, following presentation of a small high contrast visual stimulus, the response just goes up and then it comes back down with the activity always decaying monotonically with distance from the point of input. So the point of thalamocortical input in this experiment was right here. I'll play this one more time. So you can see just like the stationary bump of activation, it just rises up and then it comes back down. So I'll emphasize here that this trial averaged picture was thought to be the standard evoked population response in Cortex. But we had a sense that something different was going on at the single trial level. So to go beyond trial averaging, we developed a method to track spatiotemporal patterns moment by moment in single trial data. And using this method, which I'll explain in detail in a few slides, we could directly detect that waves were systematically evoked on each trial, as plotted here for an especially clear example. So I'll play this again. And what you can appreciate in the movie is the thalamocortical input is here, and then you can see a wave of activation spreading out from that. So now with these computational methods, we were able to reveal a qualitatively different form of stimulus evoked activity on the single trial level, where the signal to noise ratio is now a lot lower, and where this sweep of activation could dynamically influence neuronal excitability as it propagates across several millimeters of the retinotopic map. Again, this is during the awake state and was previously not known to occur. But if these evoked waves are really going to play a role in normal waking visual computation, we first had to ask an important question, and that was, do traveling waves actively affect visual processing? This is an important point because an alternative hypothesis could be that the waves could just be a consequence of the very dense connectivity in Cortex, and the visual system might want to ignore them. So to address this question, I developed a collaboration with the lab of John Reynolds at the Salk Institute, where Zach Davis had recently done a lot of work to develop the marmoset as an experimental model for primate vision. And the marmoset has the advantage of this smooth, non-gyrated cortical surface. And the experiments Zach did represent some of the first Utah ray recordings in an awake primate. These Utah ray recordings are targeted towards the superficial layers of cortex in area V5 or MT, which handles visual motion processing. And in this collaboration, our goal was to ask whether spontaneous or internally generated waves exist in the visual cortex of awake animals, and if so, whether they affect perceptual sensitivity. So now, when we looked at the spontaneous activity in these recordings, we immediately saw that the LFP dynamics were far from any kind of narrowband rhythm. They have complex fluctuations that are changing frequency and amplitude rapidly from moment to moment. This is an example LFP from an electrode on the electrode aligned to the target representation in cortex. And you can appreciate here that there are these complex fluctuations that are changing from moment to moment. So we could try to look at these fluctuations in a specific frequency range, here by filtering in the 8 to 13 hertz alpha range, for example. And while there may be some times where the power increases and fits somewhat, overall, it's not a great description of the spontaneous activity. And even here, during this more beta band epoch, there are still times where the filtered version does not perfectly match the broadband signal. So in general, we appreciated the need for an analysis technique that could be applied generally across all the individual trials and the experiments during these fluctuating complex spontaneous dynamics. And this was important to us because when we're considering phase offsets, we're now going to consider many phase offsets across many electrodes in space. These small phase distortions here, so the trough here is not matching up with that, and there, those can really add up. So much better then maybe to use a wideband representation, which is this smooth curve here, representing signal frequencies from 5 to 40 hertz, where the fluctuations in the signal can now be well described. So we developed an approach to these wideband data, which we call generalized phase. It's indicated by the color of this curve here and represented on this curve here. And it's inspired by the original analytic signal paradigm introduced by Denis Gabor in 1946, along with some numerical guarantees for the validity of the phase representation of the signal and fixes for when this breaks down. So while approaches like the Hilbert transform in general can only be applied in the case of narrowband signals and break down quickly when the signal becomes too wideband. Here we developed a set of techniques in the complex plane to represent these non-stationary wideband fluctuations in a robust manner. So what you can think about this doing is capturing the phase of the largest fluctuation on the electrode at any one point in time, again, while minimizing waveform distortion. And because of the 1/f structure in the signal, this will generally be the lower frequency components, though, as you can see here in this example, not always. And now we're going to use this approach across many channels to detect spatiotemporal patterns in the wideband data. So to illustrate how we systematically detect arbitrarily shaped traveling waves when we're considering many of these time series on an imaging array, I have a demonstration movie for the algorithm, for the wave detection algorithm we introduced. So in this movie, there's just noise at the start. And here the algorithms guess for the centers jumping around randomly. But when a simulated expanding wave appears, the algorithm finds the drifting center and reports wave detection. And when in a few moments the data return to just noise, we again get no detection from the algorithm. So what we have here is an algorithm that's robust to unstructured activity while also being sensitive to coherent flows on the imaging array. Just to briefly explain this calculation using one frame from the movie, the idea is that we take a statistical approach to traveling wave detection. So first, the putative wave center is found in each moment using a vector calculus operation. And then with the center found, we can test for a linear relationship of phase with distance from the source in the noisy and unsmooth data. And this characterization allows us to characterize the strength of the spatiotemporal pattern at each point in time by a single number, the circular linear phase correlation with distance, or rho phi d. So rho for correlation, phi for signal phase, and d for distance from the putative source point. Here, just the Euclidean distance on this regularly sampled 2D electrode array. We can then introduce a hypothesis test on rho, which takes into account the chance or background correlation in the data. And so why is this type of approach important? First, it requires no spatial smoothing, which can create artifactual waves in the data. Second, it's an improvement on previous methods, such as the space frequency singular value decomposition, which tend to break down rather quickly under noise. And third, it provides this quantitative moment-by-moment test for traveling waves and high noise multi-channel data. Now, with this approach, now we wanted to apply this technique to detect waves on the Utah array during normal vision in the marmoset. So the marmosets here were trained on a challenging visual detection task performed while recording with these Utah arrays. The task involved establishing fixation at the center of a presentation screen, and after a variable delay, the appearance of a faint motion stimulus. And finally, a behavioral report with a saccade to the stimulus location. And importantly, stimulus contrast here is adjusted so that the animal makes approximately 50% correct hit trials and 50% misses. So now we analyze the activity on the multi-electrode arrays in the moments just before stimulus onset. And we detected that the ongoing fluctuations are in fact often spontaneous traveling waves. So this here is the electrode aligned to the target representation in Cortex. This shows time in milliseconds. And the LFP here is plotted in color with pink colors for negative potentials and blue colors for positive potentials. I'll note here again that this is amplitude in the wide signal bandpass, which has this advantage of minimizing waveform distortion. And it's important to note, in addition, that the waves detected in the LFP here are traveling at the same range of speeds as those we had previously observed in the VSD signal, providing confidence that these results don't arise from particularities of any one imaging type. Finally, I won't go into this during the main talk, but similar LFP waves also occurred during normal viewing of naturalistic visual scenes. So after observing these spontaneous waves in the data, the first thing we were interested in was their speed, or how fast they're propagating across the imaging array. And we observed here that the distribution of phase speeds falls consistently between 0.1 and 0.6 meters per second. And this range was really interesting because it falls right within the range of conduction speeds for the long range, unmyelinated horizontal fibers that project across single regions in cortex. So we hypothesized then that these horizontal fibers might provide the underlying network mechanism for these spontaneous waves. And to test this hypothesis, we studied a large-scale spiking network model with excitatory and inhibitory neurons operating in the balanced regime, arranged on this two-dimensional sheet, and then connected with local random connections. Connections in the network were distance dependent, following a Gaussian with distance. And the axonal time delay between two neurons grows linearly with the distance between them, modeling the transmission of the spike down the axon. The other neurons in the network are also connected following these two rules. And now time delays are often not considered when modeling spiking networks. And in our work, we found this microscopic property has a profound effect on shaping the dynamics into traveling waves of spiking activity propagating over the network. I'll note here, this is an example from a 1D network, just for visualizing the spike trains easily. And I'll show an example from a 2D network on the next slide. So plotted here is a raster of spikes over 800 milliseconds from 80,000 excitatory neurons in the model. The inhibitory neurons aren't shown here. And the spiking here is just driven by self-sustained activity without external input. And we find here that the time delays shape the activity into these lines in the space-time plot, representing waves traveling through the system. And importantly, even though the dots may appear large here, only very few neurons are spiking as these waves go past. So through custom large-scale simulation code, we were able to find that networks of 100,000 to 1 million neurons admit a new activity state, which we called sparse waves. And the key detail here is that the network remains locally in the asynchronous irregular state or highly disordered state where neurons fire at low rates. Here it's 5 hertz, and they have very weak correlations. And to come back to our analogy from the start of the talk, if we look at only one local point in the network, we only see disorder here. And it's by zooming out to the scale of hundreds of thousands or millions of neurons that are the scale represented on the Utah arrays that we're looking at in the experimental data. There we can see waves in these networks with local activity that's consistent with the background state in awake animals in vivo. So these sparse waves were a critical insight from the model because they show how traveling waves can propagate through these systems largely in the subthreshold without introducing spiking correlations that might be harmful to perception. Lastly, I'll note that these sparse waves stand in contrast to waves in smaller networks, which tend to produce waves that turn all the neurons on or off at the same time, leading to states that are not quite as consistent with the decorrelated state typically observed in vivo. Now, we can also simulate the model in 2 spatial dimensions. And from the synaptic currents at each point in the network, we can calculate the resulting LFP using a well-accepted estimation method. And we can then visualize what the traveling waves of sparse spiking activity look like in the simulated network LFP. And so these results provide a good example of how we can connect our computational analyses in the data, here in the context of Utah arrays, to network mechanisms through these large-scale spiking computational models. And the model also provides 2 specific predictions that we can then test directly in the Utah array recordings. First, locations with relatively negative LFP will have more spontaneous spiking. For convenience, we'll call these depolarized states in what follows. Locations with positive LFP will have relatively less spontaneous spiking. So relatively positive LFP will have less spontaneous spiking, and we'll call those hyperpolarized states. So this first prediction comes from the fact that the waves exhibit changes in the EI balance at different phases of the wave, which then weakly modulate the background spontaneous spiking, but can in turn have large effects on evoked responses by changing the subthreshold state in the local networks as these waves are going past. So leading from this point, the model also makes a second prediction that these depolarized states will exhibit higher responses to incoming inputs. And we can see this by stimulating the network model during a depolarized or a hyperpolarized network state, and then measuring the response. So this plot represents a set of simulations of the network model with precisely the same initial conditions and precisely the same connectivity, with no stimulation, that's this line here, with stimulation during a depolarized state. And for example, at a point in the network here, if we sent spikes to that point in the network, and with stimulation during a hyperpolarized state, for example, at a point in the network there. Now, to test the first prediction from the model, when we quantify spontaneous spike rates as the waves propagate over the Utah array, we indeed find that ongoing multi-unit spiking during detected traveling waves is indeed higher at plus or minus pi radians. That is at the negative potential, or what we call the depolarized state, and it's lower at 0 radians. That is at the positive potential, or what we call the hyperpolarized state. These red dots here are a control, showing what happens if we consider only the phase of the averaged LFP across the electrodes. And so this observation verifies the first prediction from the model. Now, motivated by the second prediction from the network model, we hypothesized that the alignment between the spontaneous traveling waves we'd observed and incoming spikes driven by the target could affect the magnitude of the evoked response in MT. So that for a given number of incoming spikes to the cortical area, if the wave is in a less excitable state, fewer output spikes will occur at that retinotopic point. However, if stimulus-driven spikes arrive in line with the depolarized state of a wave, the same input will now drive a much stronger response. So now we quantified the distribution of phase at the electrode aligned to the target representation in Cortex during detected traveling waves across both hit and miss trials. And to do this, we used a measure that's similar to the Kuramoto order parameter in physics, which measures synchronization of coupled oscillators. So here, R sub k is the phase alignment measure. It's a complex number, and we'll work with its modulus or absolute value. N sub t is the number of trials in each condition, hit or miss. And phi is the phase of the stimulus electrode at trial j at time k. And finally, we'll put R in z-score units relative to a matched uniform circular distribution, allowing us to correct for finite size effects. So now we calculated this measure for both hit and miss trials. And this plot shows our phase alignment measure, calculated, so our phase alignment measure here on the vertical axis, calculated across hits and misses around the time of stimulus onset. And here we observe a significant phase alignment for hits during the target evoked response, as predicted. This line represents the 99% cutoff from a shuffled null distribution. But what was unexpected was that the strongest phase alignment occurred here, prior to target onset. This couldn't be explained by target evoked activity because the response only starts at about plus 70 milliseconds for these faint visual stimuli. So we called this the peak of phase alignment, or PPA. And we then developed a further hypothesis that this PPA represented alignment of the spontaneous traveling waves we'd seen prior to target onset, which then puts the local network into a depolarized state during the target evoked response. So then we conducted 2 analyses to test this further hypothesis. So first, we tested whether the phase of the spontaneous waves at the time of PPA can modulate spiking activity during the stimulus evoked response. And to do this, we selected trials where spontaneous traveling waves put the target aligned electrode into a depolarized or a hyperpolarized state at the time of peak phase alignment, or PPA. So either the system is in a depolarized or hyperpolarized state at this time. We then looked at average firing rates across the trial in these 2 conditions, and we saw that the depolarized states had significantly higher firing rates than the hyperpolarized states during the target evoked response. So this shows that spontaneous waves can modulate evoked firing activity, again, putatively by setting up a depolarized state into the future. It's important to note now that this calculation was only for the wave trials. That is where we detected a traveling wave on the electrode array in the moments just before stimulus onset. And in the non-wave trials, this effect disappears. So there's less predictive power into the future when you're not in a spontaneous traveling wave state. Second, when we compute the conditional probability of a hit given a certain phase, expressed here as the circular distance from the most predictive phase, we found that the phase of the spontaneous waves at the time of PPA, before stimulus onset, modulates the likelihood of target detection by roughly 30%. So just to summarize this section on detecting and characterizing waves in visual cortex, our computational technique can reveal stimulus-evoked propagating waves that are robustly present in single trials, even though they're obscured in the trial average data. Small visual stimuli consistently evoke radial traveling waves in primary visual cortex of the awake primate. These results show that our image processing algorithms can reveal previously unknown features of cortical dynamics. And finally, spontaneous traveling waves travel across the retinotopic map and can affect sensory processing in awake animals. Yes.
So
of course,
I've been
yeah.
meaning to ask, and
No, that's a great time to start.
This is small visual stimuli, but I've been meaning to ask you, what's the size of the stimulus with respect to the size of the array, receptive field sizes in this area?
That's a great question. So here, and I'll just use the trial average to show. So the visual stimulus here is a Gaussian point of light with a sigma 0.5 degree in visual space. So it's fairly small, and the best way to think about it is that the feed forward imprint is what we can calculate as like relatively small. And so we can definitely see that even on the trial average level, this bump of activity is going past the feed forward imprint, consistent with some of the work from Grinwald and others.
What could be, so how many neurons are we talking about? How many receptor fields? And just curious about the size of the receptor field with respect to the stimulus.
Yeah, that's a great.
Are those classical receptor fields effects, or are those already external?
These are going extra beyond the classical receptive field, right? So then these types of interactions, they go across an appreciable amount of visual space. So here, I believe the stimulus was at a small eccentricity, not foveal, but not very peripheral. And so the receptive fields here are relatively small relative to this. And this is actually, so this is about 6 millimeters of cortical space there. So it's a large section of cortex, visual cortex. Yeah, that's a great question. Yeah, so this original study was stimulus evoked and in V1. And in the second study that I showed here, this one was an MT. So we're in two different cortical areas. In one case, we're studying stimulus evoked activity, and here we're studying the spontaneous. But, yes.
Were the monkeys, the arms with moving their eyes, or did you have eye tracking? Was there movement that was creating these waves?
That's a great quote. We looked for that and we didn't see any. So this was a fixation task. So the sorry, let me just get there. Yeah. So this is really the marmosets had to maintain fixation within a narrow window. And we also looked for, because Chris Pack's lab at McGill has seen that visual saccades can create waves in this area. Interestingly, the visual saccades create a very stereotyped wave from fovea to periphery. And so we very carefully controlled that we weren't seeing those waves in the data, in addition to making sure that the eyes were in a small window or in a tight fixation window. So here we don't think. Exactly. Yeah, it's a faint motion stimulus at about 5 degrees eccentricity. And then they had to report whether they saw it or not. And then the contrast was down regulated so that it was very, very challenging and 50% correct.
Sorry, but you just mentioned spontaneous activity, so you're looking at the activity before the stimulus.
Yes, yeah. So here we're looking at the kind of the effect of the spontaneous on the evoked. They have the fixation, but there was no, there was a flat hazard function, so they had no information about when the stimulus would appear. Yeah, and that was controlled for. Yeah, sorry. No.
So if you try to average by instance,
Mhm.
then you get the way you're showing us this, time plot looks all like that inverse specific colon, but I show a time plot that I'm expecting it tuning, but is it just such a weak effect you get? There's a wave in different directions, either in times with cancels, but it's all about the days.
I see. Yeah. Well,
You average this on this is what you get a regularized, not really, but you get a picture.
that's a great question. Yeah, yeah. So this, for example, was the target aligned electrode.
Right.
And if we average there, we can see an LFP, a relatively negative LFP there. And that drops off in space pretty quickly. Yeah. We don't see a wave, and that's because the waves there are traveling in very different directions.
Waves don't any direction.
Exactly. Yeah.
So if the brain was trying to allocate this to help it out,
Yeah.
It's just all about, I don't care where it starts from, it's just gotta get to that spot at the right moment.
Yep. We've looked at that a little.
I'm sort of closing and this not for your question, you're about to get to it, but it's an interesting, if it's a useful computational thing. But it could originate anywhere within what otherwise seems like a really important enough, but now seems like an unimportant one. But or seems like it's just make sure this thing collides with the right moment.
Yeah, it's a great question. And so here in the context where the animal had no information about when the stimulus would appear, that's, so we're kind of getting that. And then the kind of follow-up that we're thinking about is kind of towards exactly that point, because now if the animal has information about when the stimulus will appear, there'll be an increase in behavior. And then also too, can we find that the waves are also linking up in the same way with that link of behavior? But here we really wanted to study the spontaneous case and just eliminate any kind of link between the spontaneous activity and the evoked, because that might have been a confound. Yeah. No, it's a great point. Yeah. But really a lot of these were going towards exactly kind of controlling for making sure it was spontaneous.
Would be a bit of a twisted question.
Sure.
So presumably this wave, reflects some kind of functional mechanisms, or it's the expectation that it should help pass a volley of spikes coming from the stimulus. So I'm just wondering whether you looked at the relationship between the speed of travel and some kind of time window that would allow to select essentially something like the other. Does my question make sense?
Yeah, of course. Yeah. I mean, and so definitely people, so Sabine Kastner, for example, has studied this kind of, this kind of theta frequency attention modulation. And so this is on roughly the same time scale. We don't know whether those two are linked, but we're kind of interested in asking exactly that question now. But here we really, so this kind of opens up these questions about what's the direction, what's everything, happening there. But I think the important thing is that we can see that the waves are having this functional role, specifically by upregulating or downregulating the gain at one point in cortex. And that was kind of the central thing that we found here, was that, the visual system might just try to actively throw these things away, but we can see at least that they're regulating gain in some specific way, and that's having a functional or meaningful impact on this, stimulus detection task, which is kind of, the very first start of understanding their role in vision.
So given the size of the stimulus, how much time and the speed of travel, what's the time window for knowing that the animal doesn't know the set of the stimulus?
Yep.
How much time is there for the spikes to go through within say one of the peak of the wave?
That's a great question. So then, so for example, I think the largest effect, so from what I've seen in the analysis, I think the largest effect is when the peak, or when this depolarized state aligns with what would be the onset transient. we haven't really figured that one out yet, but that is consistent with work from, for example, like Macknick and Livingstone, where they've really found that the onset transients are the key things for crossing the threshold of conscious perception. But that's kind of where we're thinking about going with that. And so it's really is important. Well, the alignment between, I think that we've found that these waves are kind of modulating even before stimulus onset, but I think the most important thing is the alignment with the onset transient. But that's kind of what we're going to do in future work. I want to switch now, just because we've been thinking about what computations these waves could do. After seeing that they exist in the awake state, and then also that they affect the excitability and can regulate gain and also perceptual sensitivity. It raises the possibility that, like we've been discussing, that these waves could be used for some sensory computation in the visual system. But if that's the case, we need to ask what computations these could be in general. And again, these traveling waves seem to at first belong to a really different paradigm from the standard ways we think about processing visual inputs, from these kind of feature-based models to deep neural networks. And so to understand what computations these waves might do, we focused on recurrent neural networks where inputs project with feed-forward connections into a recurrent layer here, where nodes are very densely interconnected, approximately following the pattern of connectivity in visual cortex. So they'll have local connections and time delays in a way that we can approximate the connectivity in visual cortex. And here we focused on training a special kind of network, one where the state of each node is described by a complex number. We did this to make use of a particular mathematical framework my lab's recently developed for this type of network that allows us to understand the dynamics that will result from a particular connectivity pattern in an analytical manner. So we call this network model a complex valued neural network, or CVNN for short. And so now we feed individual frames from a movie into the network, where the recurrent connections then generate sophisticated spatiotemporal patterns of activity in the recurrent layer. And now, as we might expect here, when we stimulate the network with a point source, it's going to produce a wave of activation traveling outward from the point of input via the recurrent connections. So now these recurrent connections are going to have an effect across the recurrent layer. And because these waves unfold over space and time as they propagate across the retinotopic map, we reasoned that they could provide a neural mechanism mediating short-term predictions in the visual system. So with the CVNN, we first asked whether, given an input frame from a movie, the network could be trained in a one-shot manner on that individual movie to accurately predict the following input frame. So to perform this task, we focused on a learning rule that requires training only a linear readout of the recurrent layer. And this procedure is analogous to a complex valued implementation of the reservoir computing paradigm that's recently proved very powerful for predicting chaotic systems in physics. However, this training paradigm, to the best of my knowledge, has never before been applied to naturalistic movie scenes. So now with this training paradigm, the network can get so good at predicting the next frame of the movie that the network's own prediction can now be provided as input in place of the original movie. So we'll term this process closed loop forecasting of movie scenes. And now with a CVNN trained on this way, we could ask whether the network could predict several frames into the future. So in the movie I'll show here, this is the ground truth movie. This is the forecast from the network at an optimal level of recurrence. This is the activity in the recurrent layer, and this is the forecast in the network without recurrence. So here we start by feeding the network the first half of an input movie, and then ask whether it can perform closed loop forecasting on the rest of the movie. And now with this setup, we can see that the CVNN can predict the input movie well. Importantly, I want to emphasize here that the network's no longer seeing any frames from the input. Rather, the network's generating these predictions from its internal connections alone. And this prediction stands in contrast to what we see without recurrent connections, where the network produces an activity pattern that represents only the average of the feed-forward stimulus imprints without having learned the underlying spatiotemporal process. So the real visual system operates on richly textured and naturalistic visual scenes. And we can now ask whether this same process will work for naturalistic video inputs on next frame prediction and closed loop forecasting. So to do this, we use simple naturalistic movies from the Weitzman human action dataset. As before, we trained linear readout weights of the CVNN on individual naturalistic movie inputs, and then tested whether, given the first half of the input movie, the network could produce the second half in a closed loop forecast. And now, even with a much more sophisticated input than the previous example, we find the CVNN can be efficiently trained on these naturalistic movie inputs. So here again, this is the ground truth movie. This is the forecast from the network at an optimal level of recurrence. This is the activity in the recurrent layer. And this is the forecast in the network without recurrence. And as with the previous case, the network accurately predicts the natural movie using only its connection weights. In this case, the recurrent connections in the CVNN create complex spatiotemporal patterns here, resulting from the combination of the feed-forward input and the recurrent connections. And we can see here again that the recurrent connections and the sophisticated spatiotemporal patterns they create are important for success in the closed-loop forecast, as the network without recurrence does not produce an accurate closed-loop forecast. So we can ask, what is it that's really causing or underlying the closed loop forecasts here? Is it the recurrent connections or the complex spatiotemporal patterns they create? So to answer this question, we started with networks that achieve accurate closed loop forecasts and then randomly shuffled their connection topology. That is, we took their local connections and their time delays and randomly rewired them. And now we can see that the shuffled version of the CVNN does not produce accurate closed loop forecasts, even after retraining. So this network's been retrained, but it doesn't produce an accurate closed loop forecast. So this control demonstrates that the specific pattern of connections in the recurrent layer, which again approximates the local connections and time delays in single regions of the visual cortex, are important to generate these accurate closed loop predictions. So in our paper on spiking networks in 2021, we showed how local connections and time delays on the scale seen in these single regions of visual cortex create spontaneous traveling waves over very large scale spiking neural networks. And here we're showing that these types of connections and their time delays might serve a functional role to structure neural activity into these spatiotemporal patterns that could enable quite powerful predictions. So to summarize this section on the potential computational role for traveling waves in visual cortex, a CVNN can learn to forecast naturalistic movie inputs from its internal weights alone. Closed loop forecasts depend on recurrent connections and the sophisticated spatiotemporal patterns they produce. And these results may provide a new way for thinking about computations with traveling waves in visual cortex. While their general computational role has remained unclear, traveling waves might make sense when forecasting complex and dynamic natural visual scenes a little bit into the future. Yes?
So in the first part, you're emphasizing more the role of single waves.
Yeah.
Just a single source. In this second part, you have interference patterns of thousands of waves.
Yep.
Right? Could you articulate this a little bit more?
That's a great question. So here, like you note in this section, so in the first part, we had, a simple point stimulus, kind of pinging the system and seeing what will happen to it, right? And so you get this impulse response. It's a wave propagating across. And that's a very simple stimulus. And we thought a lot about, well, what would that mean for some actual naturalistic stimulus? And so I think about this as a way to, in this case now, the input's much more complex, right? You have this naturalistic movie coming into and hitting the recurrent layer every time and interacting with the previously generated recurrent dynamics. In this, and part of the reason that we use this complex valued neural network was we can actually demonstrate that these recurrent these patterns created by the recurrent connections can actually be represented as a sum of traveling waves, because we can know that from our analytical approach to this network. So in this way, we can kind of really go from a very, very simple visual stimulus, like this just point of light, to something more naturalistic using this analytical approach. And that approach has to do with connecting the eigenspectrum of the adjacency matrix describing the system to these nonlinear dynamics in the recurrent layer. I hope that addressed your question. Thank you.
How did the delays relate to the timing? Like how is the speed of the motion that you can reproduce fixed by the speed of the delays you have, or how do you think about it that the actual frame?
That's a great question. Yeah. So for example, if we change the delays, we can make the predictions worse, right? So if we make the delays much shorter or much longer, we can make the predictions worse. So there is an optimal delay across this very simple retinotopic map that we've created.
Optimal for the movie.
Yep. Exactly. And so, or like the speed of motion in the movie. One control that I didn't show here was that we we took this network at its optimal hyperparameters and we tried to retrain it on a phase shuffled version of this movie. So that, keeps all of the amplitude components the same, but then shuffles all the phase components so that you have a kind of scrambled movie. And in that case, the network at the optimal hyperparameters couldn't learn that movie. So it goes towards showing that these networks are picking up on the kind of spatiotemporal correlations in the input movie and utilizing those, but it's just a first step. Yeah, I do. So so these waves going over visual cortex are propagating over the cortical area. So in MT, for example, it's tens of milliseconds. And I think we haven't shown this yet, but I think that that's an interesting time scale for short-term predictions in the visual system. So we know from work like from investigators like John Murray and Xiaojing Wang that, in the sensory cortices, you have relatively short autocorrelation times. And that those actually make, or they match up relatively well with what we're seeing here.
The biggest word corollary is a little deeper, not the corollary of this, that there's like a natural C and the optimal speed or something, because there's a map of the wave.
Yep.
So does the natural C that a marmoset experiences in terms of emotions, does that match the propagating wave? In other words, you want the default condition to really wave steps, but how you
Yep.
that one.
That's a great question. I mean, so we have thought a little bit about naturalistic movies for the marmosets, but controlling for all the conditions there is, and so, but we have actually thought about producing that stimulus set, and that would be exactly the type of thing that we could study.
In the simplest form, it was the simplest saying.
Yep.
Could imagine, of course, the spot capability, natural, best velocity, and
I will mention that, this is kind of all under the approximation that there's one single horizontal speed, but we know that there's actually a distribution of horizontal speeds from 0.1 to 0.6 meters per second. So we're not kind of considering that additional complexity yet. Yeah. So thank you for the questions. I have a little bit more material. I want to present that if that's okay. I know that we're in good time. Like, okay. No, it's good. Yeah. And so I have 10 more minutes or 5 to 7 more minutes. So I wanted to show this because this is another kind of wing of research that we're doing in our group. And so it's a very, very different scale and it's a very, very different problem. So we're looking at sleep rhythms related to memory consolidation during sleep. And this work builds on findings initially made during my postdoctoral research and that we've now extended in my independent research program. So just as an introduction, you may all be familiar with the two-stage model of memory consolidation, where memories are first formed in the hippocampus as a sort of short-term store, and then transferred to the neocortex by driving sparse sets of cells with ascending projections. And with the hippocampal activity driving these patterns in cortex repeatedly, the memory patterns can become consolidated so that there's strong links between the assemblies in cortex, which are then relatively independent from the initial hippocampal representation. So there's growing evidence that the rhythms of sleep play a key role in this process. And across the range of non-REM sleep, there's a set of different rhythms. So in this work, we were immediately interested in stage 2 sleep, which contains the 11 to 15 hertz sleep spindle oscillation, which is one of the most prominent in the sleep EEG. And we were specifically interested in sleep spindles because they've long been thought to be involved in sleep-dependent memory consolidation. Relative evidence has long shown that more spindles during sleep after a learning task predicts better post-sleep memory performance. And recent pharmacological studies in humans have provided causal evidence that sleep spindles help memory consolidation. So these previous studies tell us that spindles are related to memory consolidation, and we know they represent rhythmic input modulations and spiking in the cortex. But again, if this is what's happening at one point in the cortex, what's happening simultaneously across the cortex, and how could that help the process of memory consolidation? So to answer this question, we looked at spindles in human subjects recorded with intracranial ECOG arrays, which are sets of electrodes placed directly on the cortical surface as part of an electrophysiological mapping procedure for treatment of epilepsy. And these patients sleep for several nights, up to 2 weeks, while waiting for a seizure to occur. And we can then analyze the relatively normal nights of sleep. And now when we scanned across individual oscillation cycles using our computational algorithms, we found that these individual spindle cycles are organized into a specific spatiotemporal pattern on the whole brain scale. So in this movie I'll show, each dot here is an ECOG electrode. It's plotted directly on the cortical surface for a given subject. Normalized potential in the spindle frequency band is plotted in color. The red dot here marks out the time course for the channel in black here, and the other ECOG channels are plotted in gray here. So when looking at these spatiotemporal patterns, we appreciated that they formed these repeating rotating waves, traveling from the temporal to parietal to frontal lobe and onwards in this rotating pattern. So this is 5 cycles of a spindle oscillation in a sleeping human subject. There's no spatial smoothing here. This is just an activity pattern analyzed by our algorithm. And so after seeing this, the first thing that we looked at here was the speed of these waves. And we observed that the distribution of phase speeds consistently peaked between 2 and 5 meters per second. So that's almost an order of magnitude faster than the waves we found in visual cortex. And these peaks between 2 and 5 meters per second were really interesting to us because these fall within the range of the conduction speeds for the long range cortical association fibers, which are myelinated. So they're conducting the spikes much faster, but going over much longer distances. And they're also in the range of these shorter or arcuate association fibers, which have been estimated to be among the most numerous of the cortical tracts in the brain. So this suggested a possible macroscopic network mechanism for these spindle waves. But before thinking about this mechanism, we first wanted to consider how these activity patterns might aid strengthening distributed networks across the cortex through modulating strengths of synaptic weights. And a mechanism that was discovered towards the end of the 1990s that you may be familiar with is spike time dependent plasticity, where the timing of a postsynaptic spike and a presynaptic input critically determine the direction and strength of synaptic weight change. And so experiments found that the change in synaptic strength between 2 neurons exhibits a piecewise exponential dependence on the time between the presynaptic input driven by the spike in cell 1 and the spike in cell 2. So that if the input from cell 1 occurs just before the spike in cell 2, then we get a pre-post pairing, which leads to a strong increase in the synaptic weight between these 2 neurons. But if the input arrives after the postsynaptic spike, we get a post-pre pairing, which leads to a strong decrease in the weight of their synapse. So now we have a way to change weights between synapses. And we can think about how we might build strong connections or assemblies with this mechanism, where across the cortex, axonal conduction delays between two anatomically separated points can be 20 or 50 milliseconds. And here there's a problem, because if spindles were perfectly synchronous across the cortex, then if spikes were emitted in region A and region B at the same time, at the peak of the spindle population rhythm, then the EPSPs caused by connections originating from A would arrive at B with their axonal delay, leading always to a pairing within the window for depotentiation by SDP. And eventually, with enough repeats, the networks would in fact become decoupled, as has been pointed out by theoreticians several times since the initial discovery of SDP. But if instead, now you have this flexibility, so spindles can be self-organized into these large-scale wave-like patterns, with phase speeds matching those of the underlying fiber networks, then EPSPs caused by connections from A can align with the local burst of population activity in B, creating the conditions necessary or so that synaptic potentiation can also occur. So what I'm showing here is highly simplified, but similar kinds of ideas have been discussed going all the way back to a paper from Caltech in 2008. And further, in a theoretical paper from 2011, we in fact provided analytical mathematical solutions to the expected weight change for SDP and neural oscillations in these cases. So we can generalize this type of approach much more. And now the key prediction here is that the phase offsets of spindles at different points in the cortex may be a circuit mechanism for memory consolidation. And to test this prediction, we've developed a great collaboration with the clinical teams at Western, where we're analyzing depth electrode intracranial recordings, or IEEG, taken in the epilepsy monitoring unit, alongside scalp EEG from healthy human subjects. And in recent work, we've developed new techniques for waveform detection using machine learning methods adapted from tools to detect earthquakes and gravitational waves. And we're finding that widespread multi-area spindles occur much more frequently across the cortex than previously thought. And so now in a new set of work, we aim to test how different memory conditions during wake lead to changes in spindles during intracranial recordings. And to test this, a team from my lab, including a neurologist who took a research year to learn about computational neuroscience, is now administering memory tasks in the epilepsy monitoring unit while patients are being recorded with intracranial electrodes to pair visual scenes with arbitrary sounds. We then relate later memory performance to sleep spindles analyzed with our computational algorithms at one of the highest spatial and temporal resolutions currently possible in the human brain. So I think this is a really exciting place where computational neuroscience can come together with data to understand something fundamentally new about the mechanisms of memory in the human brain. And tomorrow I'll describe some of the preliminary data we've recently collected to show that the spindle waves change dynamically during a nap following learning. So with this, I hope you've seen several examples today of how we can go from analysis of cortical dynamics to network models and theory to capture the underlying phenomena, and then back to collaboratively designed experiments and analyses to test model-driven predictions. So thanks again, and I'll take your questions.
Yeah, maybe I missed it in the last part.
Yeah.
Maybe it seems that you're emphasizing cortical interactions during those stimulal waves.
Yep.
But could you say something more in terms of the cortical interaction with the generations of those spindles, and then how would that fit with the memory consolidation?
Yep, that's a great question. So we've seen that we have these phase offsets in the spindles, and what we're thinking about is that they, the mechanism that we've been thinking about in some of the oscillator work that we've been doing goes exactly towards this, is that the spindle kind of the thalamus kind of sets the rhythm for the cortex. And we know that the spindle is generated by these thalamocortical cells interplaying with the reticular nucleus cells at some very specific frequency. So the thalamus kind of sets the rhythm for the interaction. And then the cortical fibers shape the spatiotemporal dynamics of the spindle in cortex. So the thalamus kind of sets the beat, and then the cortical fibers are shaping the spatiotemporal dynamics. I'll go into more detail about that as well tomorrow.
Thanks for the great talk.
Thank you.
So I'm trying to connect the first and the second half. So, I mean, correct me if I'm wrong, or if this is a different interpretation, but it seems like the spontaneous wave that facilitates perception would be an indicator of kind of like how close the brain is to a critical state in terms of how receptive it is to new input and detect something. And that would be very general, no matter what I see.
Yep.
Kind of perception. Whereas in the recurrent neural network, to me, it seemed like the spatiotemporal dynamics there were embedded in the local synaptic weights, and that it helps it to do prediction about dynamics that was trained to do for that specific task.
Yeah.
In essence, it has almost like a matched filter for that motion.
Mhm.
Whereas if you took that trained network and had to try to predict a different kind of dynamic, I wonder whether there actually could be interference, and it would, in that case, the waves or the spatial dynamics might actually
Against the,
performance.
Yep.
So are those two, like, are those different interpretations?
yeah, it's a great question. And so here, that the recurrent network really only had retinotopy, but we could imagine in future things adding like orientation selectivity, so we didn't include any of that detail that we know is in the visual system. So having patchy connectivity with orientation domains and things like that could go towards exactly what you're saying, because here it's kind of a coarse wave and that it's just encoding retinotopy But- in the recurrent neural network.
But still, the general question is just like, is the spontaneous wave in the first part reflecting just the general readiness that has nothing to do with content?
Yep.
Versus the second part where it's trained very specific statistics of the movie.
Mhm.
Those seem like different interpretations too.
That's it. That's an interesting question. We have, so in terms of the first part, we have started to look at the waves more. So the waves recorded on the Utah array, and look at them with more subtle methods to look at whether there are kind of spatiotemporal patterns that group together, using a clustering algorithm to group different spatiotemporal patterns together, under the hypothesis that we may be seeing some reflection of the patchy connectivity in MT, and trying to see whether we can infer something about waves propagating through subnetworks of motion preference. We see some indication that we can predict some motion specific, and like so actually in the task we had different directions of motion at each retinotopic point. So we're looking at that now, and we're kind of trying to bring those two lines of research together at the moment. Yeah. Yeah. Uh-huh. Yeah. So like picking out faint stimuli in a kind of complex environment. Oh, I'm so sorry, and I can't really hear you very well. Sorry. Oh, I see. Yep. Definitely. And so here, in this case, the subjects were kind of in an overtrained state. So, and so they're kind of knowing which place the stimulus was going to appear. So a generalization of this would be to have stimuli appearing at different points in the retinotopic map, or having much more target locations. And that's, yeah. So different target locations and different times of appearance would exactly go towards that point, I think. And we've thought about, how would we design a much more sophisticated paradigm, which would include something like visual search, and even in naturalistic scenes, but it's, yeah, kind of stuff that we're doing now. Yeah.
Well, there's no more question. Let's thank Lyle.
Thank you.